ASHA, Assistant Professor in DKM College, the Department of Biochemistry. Now I am going to discuss about pancreatic hormones. Pancreas, it is a leaf-like structure which is present below the stomach. 2% of the pancreatic tissues are endocrine in nature and 98% of pancreatic tissues are exocrine in nature. It is made up of 4 types of cells: alpha cells, beta cells, delta cells and gamma cells, and these cells are represented in the form of A, B, D and F. A cell, it secretes glucagon, B cells, it secretes insulin, delta cells secretes somatostatin, gamma cells secretes pancreatic polypeptide. The group of pancreatic cells are called as islets of Langerhans. And about arrangement of cells, in between the 2 exocrine cells, 1 endocrine cell will be present. Beta cells, they are present at the interior of the pancreas, whereas alpha cells, delta cells and gamma cells, they are present at the central or at the periphery of the cells. About 60% of the cells are constituted by beta cells and about 25% of A cells and 10% of the cells are constituted by D cells. Specialization of the cells, there are 2 types of junctions over there. One is tight junction and gap junction. In the case of tight junction, cells are arranged closely and there is no gap in between the cells and it has some interstitial fluid. In the case of gap junction, there will be a gap in between the cells and the cells are loosely arranged through which the transport of ions will take place. Then about structure of insulin, it is a small globular protein with a molecular weight of 5700 and it is a polypeptide hormone. It has 2 chains, A chain and B chain. A chain contains 21 amino acids and B chain contains 30 amino acids. Totally it has 51 amino acids. In the case of A chain, it has intracellular disulfide linkages at the position of A6 to A11 and 2 interchain disulfide linkages will be present at the position of A7 to B7 and A20 to B19. This is about the biosynthesis of insulin. The first formed insulin molecule is called as pre-proinsulin. It has a molecular weight of 11.5 kd with 109 amino acids. This is taking place at endoplasmic reticulum. After the cleavage of 23 amino acid leader sequences, it forms a proinsulin molecule with about 86 amino acids and 9 kd. This enters into the Golgi complex where it undergoes some cleavage processes catalyzed by the enzyme, proteolytic enzyme. There are 2 proteolytic enzymes over there, PC1 and PC2. These PC1 and PC2 acts at the specific amino acids at arginine 31 and arginine 32 at BC junction and it splits 32 and 33 proinsulin molecule. Whereas PC2, it cleaves the amino acid at the position of 64 and 65. At the 64th position, it has lysine molecule and at 65th position, it has arginine molecule and it splits the 65 and 66 proinsulin molecule to form an active insulin molecule which has only A and B chain. Along with that, C peptide will be cleaved off. After this formation, it enters into the secretory vesicle. From the secretory vesicle, it fuses with the plasma membrane and it is released into the bloodstream by exocytosis process. Then insulin destruction. In the case of human beings, injected insulin, it has the off-life of about 5 to 10 minutes and it gets circulated within 10 to 15 minutes. The major destruction pathway of insulin are receptor mediated hydrolysis. This takes place in the organelle called as lysosome. Then glutathione insulin transhydrogenase is an enzyme which is present in the liver and also it is present in lesser extent in some other tissues, catalyzes the degradation of insulin molecule. Then another enzyme is a insulinase. It is a protease enzyme. It degrades insulin in liver and kidney. Then biological actions of insulin. There are four major biological actions. One is role of insulin in membrane transport of glucose, and role of insulin on carbohydrate metabolism, lipid metabolism, and protein metabolism. First one is role of insulin in membrane transport of glucose. In this case, insulin molecule transports a glucose molecule from the blood into the interior of the cell through a carrier protein. The name of the carrier protein is glucose transporter, which is represented as GLUT by the process of diffusion, and this is taking place in the presence of a carrier molecule, so it is termed as a facilitated diffusion. There are four types of glucose transporters. One is GLUT1, T2, T3 and T4. T1 transporter, it is present in blood-brain barrier, and T2 it is present in the liver, and T3 it is present in adipose tissue, and T4 it is present in adipose tissue and muscles. In the case of GLUT2, the enzyme is glucose kinase, and in the case of GLUT4, the major enzyme is hexokinase. The mechanism of glucose transport into the cells by the insulin molecule is represented here. Whenever the insulin molecule goes and binds to the receptor which is present in the plasma membrane, it forms a complex called as insulin receptor complex, that is HR complex. It catalyzes the cyclic AMP formation. After that, the glucose transporter which is present inside the cytosol goes and binds to the plasma membrane, and it faces the outside of the cell and it binds with the glucose molecule. After the binding, it shows some conformation changes. One is conformation changes 1 and conformation changes 2. After that, the glucose transporter faces into the cytosol of the cell and it delivers the glucose molecule into the cytosol. Then the glucose transporter comes back to the original position and it binds to another molecule of glucose molecule. This process subsequently goes on. Whenever the glucose molecule is released into the cell, it immediately gets phosphorylated to form glucose 6-phosphate in order to reduce the concentration of glucose inside the cell. Then role of insulin on carbohydrate metabolism. In this case, insulin has two actions. One is direct action and one is indirect action. In the case of direct action, it catalyzes some of the enzymes like glucokinase, phosphofructokinase and pyruvate kinase, which are the major enzymes involved in glycolysis. By this process, it converts all the glucose molecules into the pyruvate molecule, hence it reduces the glucose molecule level in the blood. By indirect action, it decreases the activity of the enzyme like glucose 6-phosphatase, fructose 1,6 biphosphatase, phosphoenol pyruvate carboxykinase and pyruvate carboxylase, which are other key enzymes present in gluconeogenesis. Then it promotes the activation of enzyme called as glycogen synthase, by which the glucose is converted into glycogen and gets stored in the liver. Then it inhibits glycogenolysis by inhibiting the enzyme liver phosphorylase. Thus, by the conversion of glycogen into glucose is inhibited. Then role of insulin on skeletal muscle. In the case of skeletal muscle, this insulin has two actions. One is lipogenesis, and it promotes a glucose transporter to transport the glucose inside the cell. Then role of insulin in liver and muscle cells. It stimulates glycogenesis process by activating the enzyme glycogen synthase. Thereby, it converts all the glucose molecules into glycogen and gets stored in the liver and muscle cells. Then role of insulin on lipid metabolism. In this process, it dephosphorylates the acetyl-CoA carboxylase. Thus by it promotes the fatty acid synthesis, that is lipogenesis. Then it inhibits lipolysis process, which is taking place in liver and adipose tissue by dephosphorylation of triacylglycerol lipids. Then role of insulin on protein metabolism. Insulin, it inhibits protein degradation, that is catabolism process. Thus by it decreases the rate of amino acid release from the cell and it decreases gluconeogenesis in the liver. Then it promotes protein synthesis. Overall action of insulin is it stimulates glycolysis, glycogenesis, lipogenesis and protein synthesis. It inhibits glycogenolysis, gluconeogenesis, lipolysis and breakdown of proteins. Then about insulin receptor, it is a glycoprotein which is present on the surface of the plasma membrane. It is a tetramer formed of 4 subunits, 2 alpha subunits and 2 beta subunits. These 2 alpha subunits are present at the outer side of the plasma membrane, whereas the beta subunit is present in the inside of the cell cytoplasm. Both the subunits are linked by a disulfide bond. It is an enzyme linked receptor. It has multiple enzymes and each cell has 10 to the power of 3 to 10 to the power of 5 receptors. Then regulation of insulin secretion, about 40 to 50 units of insulin are secreted daily in the case of humans and its normal value is 20 to 30 μu per ml. These are the 3 regulating mechanisms which regulates the insulin secretion. One is chemical, then hormonal, then neural mechanism. Then effect of insulin deficiency. Whenever the insulin is deficient, this are the some of the disorders which occurs in the human beings. One is hyperglycemia, glycosuria, polyuria, polydipsia and polyphagia, weight loss, then hyperlipidemia and diabetes mellitus. This is about the role of insulin biosynthesis, etc. Thank you.